In this video, we are going to look at another group of biomolecules, and they are the lipids. And there are four groups of lipids that we will discuss as we proceed through the video. The first group are triglycerides. Triglycerides are also known as fats. So let's look at particulars of fats. Here you see pictured a Hardee's 2/3 pound monster thick burger. It looks pretty hefty. Let's look at the nutritional makeup of this burger. First of all, if you eat one of these, you are consuming 1,300 calories. Remember now, those are kilocalories. 1,300 calories out of a 2,000 calorie daily requirement. That's a lot of calories in one sandwich. If you consume this, you'll have also consumed 3,140 milligrams of sodium when about the maximum recommended in a day is 2,300. So a lot of sodium, really unhealthy to have that much sodium. It also contains 90 grams of fat, whereas the daily recommendation on fat is anywhere from 44 to 77 grams. So a lot of fat, a lot of calories. There's where a lot of your calories are coming from. And of that fat, 33 grams are saturated fat, which is the fat that is discouraged from being consumed to any great extent. So you can see here it's at least half again as much as you should be getting in a single day. So fats, they're in the foods we eat, and really extreme foods contain really extreme amounts of triglycerides. So let's look at some details of what triglycerides actually look like chemically. So triglycerides are going to be composed partly of a familiar molecule, glycerol. We ran into this when we discussed cell membranes, and we talked about phospholipids. Here you can see glycerol. You can see that it's got 3 hydroxyl groups, so it definitely would be water soluble. It looks almost exactly like glyceraldehyde, except you don't see a carbonyl group at the end, and so you would not mistake it for glyceraldehyde. But again, you can remember and tie in that those 3 hydroxyl groups will be the places where we're going to come in and attach 3 fatty acids. So you can see they're going to branch off to the side. These can be various sizes. You can see the carbonyl group in the structure of each of those fatty acids, and again we're representing them generically by using that letter R, so some chain. They may or may not be the same fatty acid. That's not relevant. So if you've forgotten what fatty acids look like, let's take a glance at a couple of them. First of all, you have what are called unsaturated fatty acids. So you look at these structures, how would you distinguish them from carbohydrates? Well, you don't see any hydroxyl groups along the length of the chain in both of these fatty acids. But at the end, you do see a familiar landmark. There's that functional group. There's a carbonyl group, which is also part of a larger group, a carboxyl group. So again, you're learning to recognize landmarks and be able to typify the character of these molecules. We also notice that in the middle of the chain, right there, you see a carbon-carbon double bond. We know carbon likes to have 4 bonds, and you can see most of the carbons in the chain have 4 different groups attached to them, except in this one position. So that's why it's referred to as being unsaturated. This happens to have only one of those carbon-carbon double bonds, and so that is why it is called a monounsaturated fatty acid. Below that, linolenic acid is a polyunsaturated fatty acid. There's your carbonyl group again. There's your carboxyl group. You've got this long chain of carbons and So carbon-carbon bonds and carbon-hydrogen bonds. So most of it is non-polar, definitely going to be hydrophobic. But this time we've got multiple locations, three locations where we have the carbons are not saturated with hydrogen, and that's why this is called a polyunsaturated fatty acid. If you remember from health, these are the preferred kind of fatty acids to have in our diet for various reasons that I'm sure you will touch on in biology next year. In addition, we have saturated fatty acids. So again, carbonyl group, part of a carboxyl group. This time all of the carbons in the chain are filled in with hydrogen, so this is saturated. Down below, a slightly larger molecule, stearic acid, carbonyl group, carboxyl group, so a little bit of polarity at one end, but mostly nonpolar and therefore hydrophobic. So we take these guys and we attach them to that glycerol backbone. Here's a specific one. This is called tristearine because it's got three stearic acid molecules. Those are the fatty acids that have been attached to the end. You can see this is getting to be quite large compared to the molecules that we looked at when we talked about carbohydrates. Highly nonpolar, highly hydrophobic fats. They are not water soluble. So again, you're learning to look at a structure and winnow through the jumble of letters and recognize landmarks. You recognize landmarks, you know their character, you'll know the character of the entire molecule, and therefore you'll have a sense of what it's going to do chemically when it is present in cells. So let's look at a meal. Here's a healthy meal. We've got potatoes, we've got salmon, and we've got some asparagus, and that's a pretty healthy meal. All right, so you eat this meal, you consume it, it goes down your esophagus, it enters the stomach, and your stomach acids and digestive enzymes start acting upon the fats that might be found, the triglycerides that might be found in that food. All right, but now again, remember the character of these triglycerides is that they are not water soluble. So that's going to pose a problem when we go to digest fats. They're going to tend to want to clump together. So you're trying to break these large globs of fat droplets and breaking them into smaller ones. But as it moves through and heads towards the small intestine, it's still kind of more of a glob than anything else. So we're going to have to do something. We're going to have to use some chemistry. Your body's going to use chemistry to attack that glob of fat if we're going to get to the things we need from it. So if we look at a little more detail of your digestive tract, here in this diagram you can see at the top the esophagus coming down and joining into the stomach. So the food you consume goes into the stomach. You've got hydrochloric acid in there. You've got digestive enzymes. You're going to start attacking any food that you've consumed. At the far end of it, you see it kind of has a serpentine kind of shape as it moves into the duodenum, which is part of the small intestine. And right there, you in the crook of that stomach and duodenum, you have your pancreas. You can see the liver is nearby. It's feeding in through the gallbladder, and this is where we're going to start attacking that glob of fat. So the gallbladder stores away bile acids, and that bile produced by the liver can emulsify the fat. And what that means is we're going to try to break it down into smaller and smaller droplets. We want really tiny droplets. We're trying to increase the surface area. We got to get digestive enzymes in contact with the fat. In your pancreas, you have a digestive enzyme specifically for fats called lipase. So that's being dumped into the small intestine to start acting on those tiny droplets and start breaking down the triglyceride into its components, glycerol and fatty acids. And if we again look at even finer view of the digestive system, here again in the small intestine, you can see those little microvilli in the lumen of the small intestine. You can see the droplets being broken into tinier and tinier pieces. The microvilli help to increase the surface area, to increase absorption. You break this down into your fatty acids and glycerol. You get them small enough then to be transported across the membrane into the epithelial cells, and then they can be put back together. They're reassembled. Now, again, these are hydrophobic molecules, so we can't just dump them straight into the bloodstream. So they form what are called chylomicrons. We attach some protein to it. It goes into the lymphatic system. You can see at the very, very bottom, you can see the lacteal, and farther along in the circulatory system, in your jugular vein, we will dump these into the bloodstream, and then they can go off to the adipose, the fat tissue in your body. Fats are great sources of energy, and here you can see one of the cycles you will learn about next year. Those triglycerides, if we need extra energy, if we've used up glucose and we need more energy, we're going to chop those fatty acids up into little 2-carbon units called acetyl-CoA. And acetyl-CoA is metabolized very much like sugars or carbohydrates were, and they go into the mitochondria and enter the same metabolic cycle that's known as Krebs cycle, or the TCA, which is tricarboxylic acid cycle, or also called the citric acid cycle. And we're going to extract energy and produce ATP, because again, cells have a sort of energy currency. ATP is the primary one. If we don't need that energy, if this is excess calories that we've consumed, then our body's going to turn it back into its own fat, and this will get carted off and become adipose tissue, so increase your fat tissue. It's called lipogenesis, so literally means producing fat. So a quick highlight then for triglycerides, fats. They are nonpolar, they are hydrophobic. They are very energy dense. You saw how big those molecules are. They're very, very large. And in all those carbons that are connected in those long chains, we can start chopping them up. Fats have more than double the energy per gram as compared to carbohydrates, 9 kilocalories per gram. So a great source of energy, but not the preferred one usually. Though we can do things, in our diet and to try to help burn off fat in the way we exercise. If we don't need it, it gets stored away in our bodies. The excess calories. Daily recommendation: 30% of your diet pretty much should come from fats. So you don't want to totally avoid fats. That's not the message that's being sent here. But excess amount of fat would be excess calories, and it's going to accumulate on your body, and you don't want that happening. So those are triglycerides. A second group of lipids are phospholipids. We've talked about these guys before. We know that these form the basis of cell membranes, and remember, it's going to offer up a barrier defining the inside and outside of a cell. So once again, glycerol is going to be the backbone. It was the backbone for triglycerides. It's also the backbone for phospholipids. Slightly different structure. What we're going to attach to those 3 hydroxyl locales. 2 of those locations are going to be fatty acids. The third one is going to be a phosphate group. So this is not 100% nonpolar. You've got that phosphate group that's charged up. That's going to form your little polar head. Remember, we form what look like little tadpoles with a double tail. Perfect polarization as far as the overall structure. The fact that you've got a polar end and a very large nonpolar end makes it perfect for forming that barrier. We need a lipid bilayer, and there you see the fluid mosaic model for the membrane. We've talked about this extensively. You know that if something is nonpolar, needs to get in the cell, it can pretty much, if it's not too big, can just kind of dissolve straight through the membrane. But things that are polar are going to need embedded proteins in the membrane, and those channels will be used by larger molecules and by charged or polar molecules. You can see there are other proteins, there are other functions that we haven't talked about in detail. You'll learn about that in biology. So that is a second kind of lipid, the phospholipids, using glycerol again as the backbone for constructing it. A third group are the waxes, and here is a wax molecule. Here's about a 40 carbon chain, and you can see you've got all these carbon, 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 hydrogen bonds. You can see this is extremely non-polar, hydrophobic, perfect, absolutely perfect for its job. It's meant to repel water. You'll see this in animal and plants employed to do just that. In a plant, what we want to do is coat the leaves with this waxy coating that's going to cut down on the loss of water from the leaves so that they don't totally dehydrate, trying to protect themselves. Birds secrete waxes onto their feathers. They're trying to make their feathers hydrophobic, and somewhat waterproof. They don't want these feathers to become waterlogged. That would weigh them down. You can imagine if this is the feathers of a duck. If this were to become waterlogged, it would soak into all those nooks and crannies. It would become very heavy and it would drown. So there is a reason for wanting to repel the water. So those are waxes, highly, again, like most of the lipids, highly nonpolar, hydrophobic. The fourth and final group are the steroids. And steroids essentially are derived, the steroids we make in our body for various functions are derived from cholesterol. So cholesterol is sometimes portrayed as we want to eliminate it. We really don't want to eliminate it from our diet. We need it as the basis for producing a variety of molecules. Here you see the basic structure. You've got these four carbon rings. Three of them are six-member rings. The fourth one is a five-member ring. There are bad effects for cholesterol. The plaques that form in blood vessels and clog up your arteries. That's a bad aspect of it. But there are a lot of things that have to be made that for our benefit. There are functions that have to be performed, and things like bile acids that we just talked about with the digestion of fats. Vitamin D has that kind of structure in it, so we can make vitamin D in our skin cells. And steroid hormones are, some of the hormones are protein in nature, but several of them are steroids, and they are made from cholesterol. So just real quickly here's cholesterol. Here's a cholesterol molecule. There you see that central four carbon ring structure that then will be repeated in anything that's going to be derived from the cholesterol, like vitamin D. We've talked about vitamin D a little bit before. We obviously can see it's a fat-soluble vitamin, important for aiding the absorption of calcium. So without vitamin D, the calcium going through our digestive system can't be absorbed, and therefore calcium plays a big role in a number of areas. You're more familiar with it being used to strengthen bones. Testosterone, a male sex hormone, again is derived from cholesterol. You can see the four rings again. So the development of secondary male characteristics are attributable to testosterone. Female sex hormones, here progesterone is a steroid hormone, and again it is development of female secondary sex characteristics. So those are the four kinds of lipids. You have the triglycerides, fats. You have the phospholipids used in cell membranes. You've got waxes that are used to coat surfaces and help slow down dehydration. And then finally you have the steroids, which form the basis of a number of different compounds.